Hi class, today we're going to be looking at an overall review of Chapter 10 to prepare for the Chapter 10 quiz. And so we'll start, if you have your notes packet, you can just kind of go through and highlight some things and make sure you have some stuff written down to indicate what would be good things to study. So 10.1 went over scientists and kind of the history of DNA. So first we had three scientists. One was Griffith, and his final discovery was that genetic information can transfer from one cell to another. Okay, so that's important in that later, as we go down through here, we'll be able to see that DNA can actually leave one type of cell and move into another type of cell and kind of take over that cell. I described it to many of you in class that were having questions on it as almost like zombie cells. So these zombie cells take over. They send their DNA into another living cell and take over that cell. Okay, 2 was Avery. And he had been the first one to say that DNA is genetic information. Okay, and then the ones that came in after him to really concrete that statement was Hershey and Chase. So Hershey and Chase used a virus, and it looked kind of like this. Okay, and it has protein, and it has DNA. And they allowed this virus to infect a bacteria, and they found that the radioactive DNA was inside of the bacteria, not the protein. So at this point they determined that DNA is actually the genetic information being passed on from one cell to another. Okay, so then next we have our next scientist was Watson and Crick. And in your notes packet, this actually starts 10.2 of your notes. So Watson and Crick actually came up with this shape being a double helix. And they confirmed what the shape was and how it was made up. And they gave this repeating units called nucleotides make up the DNA. And so you will need to be able to identify the parts of this nucleotide. So if we go back one there, and we take a look at what DNA actually looks like. So DNA stands for deoxyribonucleic acid. It's made up of nucleotides. And nucleotides have 3 parts, and you need to know this. So first is a phosphate, and then that's attached to a sugar. In DNA, that sugar is deoxyribose. That sugar changes when we go to RNA. And then it has this nitrogen base. Now the nitrogen bases, there's 4 different ones in DNA. They can be adenine, guanine, cytosine, and thymine. Now with those 4, there's certain ones that go together. So A matches with T, and C matches with G, always. Now one thing that kind of came up in one of the quizzes that we took, the little mini quizzes, is our percentages. If you were to see thymine being 40% in a cell, then we would know that adenine also had to be 40% because these match up. And so in order for them to, one of them to be 40%, the other one has to also be in a 40% range. That would leave us, that's a total of 80%, that would leave us 20% remaining, and so guanine would be 10% and cytosine would be 10%. Now, these numbers are not exact. It's not like every single time you see DNA, it's going to be 40% A. This was just one of the questions that came up in one of your little mini quizzes to check for understanding, and a lot of you had missed it. So if you see a percentage here, that percentage of thymine will equal adenine. So if we were to take out the actual percentages here, just so you don't think that that's the number that's always there, and instead we just write A is equal to T and C is equal to G. That may help you. Okay, purines are A and G, so let's use a different color here. These are called purines, and these are called pyrimidines. Okay. So now another key part, and you're really going to need to know this, is DNA replication. So this starts 10-3. So DNA replication. And the question is, why does it happen? So first, why? To make a new copy of DNA before mitosis. The when in S phase of interphase. Okay? So now the process. How does it happen? So here's the how. Number one, first we're going to show that we have these nucleotides here. These are the nitrogen bases. Just going to show kind of a little simulation of what that would look like. You have A's and T's across from each other and C's and G's across from each other. And then what we're going to have happen is we're going to have an enzyme called helicase. Okay, and it is going to unwind the DNA and create a replication fork. Okay, and that replication fork is happening right here, where it's separating from each other. So helicase comes in, it starts to separate the DNA, and now we're going to have kind of this opening where replication is going to occur. Okay, step 2 is going to have another enzyme come in, and this other enzyme is called DNA polymerase. And so we're going to show that up here. And DNA polymerase is going to come in and add new bases. The bases are A, T, C, and G. Okay? And so as it comes in, we'll show that kind of in a different color as well. As they come in, they're adding these bases across from the strand of DNA, the old strand of DNA. On the other side, it's going to happen in fragments of sets of 3. And those are called Okazaki fragments, if you remember. We talked about that in the notes originally. So our step 3 is that we have another enzyme that comes in. I'll give that a different color here. I'll do green. And this other enzyme will occur on both sides of the strand. Put that picture there. And this third enzyme called ligase comes in and fixes mistakes and links the Okazaki fragments. Okay, so this is DNA replication, and it only occurs because we're trying to make a new cell. All right, next we get into transcription translation. So transcription translation, and this is going to be the making of proteins. So first, transcription is making RNA. It occurs in the nucleus, and here's the process. We're going to have our strand of DNA. Okay, we are going to have an enzyme. Helicase does the same thing it did before. Unwinds DNA. Step 2. DNA polymerase, that was 1, 2, sorry, RNA polymerase makes RNA. So, we have here these bases, and what we're going to end up having go across from them are RNA strands. And it's going to be one long RNA strand, a messenger RNA. Now, it makes RNA. Now, it can make 3 types of RNA. So, messenger RNA, its process is to go to the ribosome to help read the code or have the code that DNA gives it. We have transfer RNA, which looks kind of like a T and carries amino acids. And we have rRNA, and rRNA makes the ribosome. Okay, so those 3 things are being made in transcription. So next we go to translation. And translation is taking, just like you think you translate a language, instead of translating Spanish to English, in this case you are translating a set of 3 letters on the messenger RNA code and turning them into amino acids or proteins. So we are translating it from letters to something that the body can use, which is a protein. So in this case, we are taking messenger RNA to the ribosome. It gets read. And transfer RNA brings amino acids to the ribosome to make a protein. So here's our step. We are going to have the ribosome. So this occurs at the ribosome. Okay, so we're going to have messenger RNA that comes and gets read in here, and then we're going to put these letters in. So we're going to put A, U, G, C, C, C. Let's do U, G, G, and U, A, and it would continue. Sets of 3 are called codons if they're on the messenger RNA. So across from these letters are going to be a transfer RNA that comes in. It will have an amino acid at the top. Okay, so the transfer RNA comes in and it will match up across the letters that are supposed to be there. So U, A, C. Now that's what it always starts with. This right here is a start codon. It means start, but it also means methionine, which happens to be the amino acid that it starts with. Now, you will not have to memorize those. You will have a chart in your book that you can use, or not in your book, on a quiz that you'll use. Okay, so we have this transfer RNA come in. It brings in the